Welcome, everyone, back for another episode of Laughter Through Tears, the 120 podcast. I'm your host, Jackie. I live in Central Maryland, have a lot of experience with chronic conditions. I have hypermobile Ehlers-Danlos syndrome, POTS, Crohn's disease, and osteoporosis. I also am a septic shock survivor, and depending on when this episode airs, it's going to be on or around the 5th anniversary of my life day on May 22nd. It'll be 5 years since I survived septic shock from a colon perforation that happened when I had a case of undiagnosed diverticulitis, an infection in my colon. And anyway, so I made it back from that. I continue to deal with chronic conditions every day. And my guest here today is going to talk about something that's very vital to our sepsis community and anyone with chronic conditions, especially those of us who need to use antibiotics, whether intermittently or on a regular basis. So today's topic is going to be very important for a lot of you, I'm sure. And I want to thank you all for tuning in again today. And my guest is Robert Donald. You might have seen his episode in the fall on vaccines. And he's a vaccinologist. He is a research microbiologist. And AMR is something that he's really had a lot of experience with. But other than that, too, he has broad experience in the biology of bacteria, viruses, and parasites. He earned his doctorate degree at the University of California, Santa Cruz, and did postdoctoral research at the University of California, Berkeley, and the University of Pennsylvania. Now, his commercial research career has been dedicated to developing new antimicrobial drugs and vaccines that can prevent life-threatening infections that sometimes lead to sepsis. Well, while a senior research fellow at Merck and Company, he contributed to drug discovery programs aimed at identifying new antibiotics to prevent bacterial, fungal, and parasitic infections. And more recently, he was the senior director at Pfizer. He led early-stage vaccine development programs, advancing vaccines to prevent a variety of life-threatening bacterial infections, including the drug-resistant ones. And again, you're going to hear a lot about that today. These can include those caused by E. coli bacteria or C. difficile, Enterococcus, and Group B Streptococcus bacteria. We've all heard of strep, I'm sure. He also managed molecular biology and immunology assay teams supporting these programs, as well as Pfizer's successful RSV and COVID-19 vaccine programs. He's now retired from R&D. He applies his previous experience to advising and educating, and in addition to serving on the Sepsis Alliance Advisory Board. He is a consultant to CarbX, a global nonprofit partnership focused on supporting the development of new therapies and vaccines to address antimicrobial resistance, or for short, AMR. Recently, he was a consultant to the Gates Foundation on antibiotic use and the monitoring of AMR in sub-Saharan Africa. In his personal life, Robert is a husband and father of 2 adult children, and in his free time, he enjoys walking his dog, practicing yoga, and swimming. So Robert is extremely busy, so I am extremely happy to have you back again today, Robert, for another episode of Laughter Through Tears, the 120 podcast. Welcome.
Well, thank you, Jackie. First of all, I'm delighted and honored to be on your podcast again to support the important work that you do covering important healthcare topics for a general audience, which is very important.
Well, that's one of the things I hope to do with this podcast too, is to help. I'm a former teacher, so I always am hoping to educate and also to demonstrate that, you know, regular patients, people in chronic illness communities, scientists, doctors, we can all come together and we can all put this information out there for everybody, especially us users of the information like the antibiotics, to more fully understand. And we can appreciate, why. I'm going to get right into it here with antimicrobial resistance and just trying to understand, what is it? And, why should we all care about it so much?
Right. No, the problem of antimicrobial resistance, it happens when bacteria not just bacteria, but mainly bacteria, evolve ways to survive the drugs that are designed to kill them, ones that we use clinically to prevent infections. And they're becoming less effective for preventing some of these dangerous infections. So I hope today we can explain why this is an important topic and delve into some of the root causes and possible solutions. It's often referred to as a silent pandemic because it lurks relatively unnoticed, while it can wreak havoc and claim lives for those that are affected. And it threatens the foundation of modern medicine. So in the golden age of discovery between, let's say, the 1940s to 80s, we had vaccines for the first time that prevented many serious infections, but also antibiotics for the first time that would actually prevent active infections. And so around the First World War, time of the First World War, we had the first widespread use of penicillins, which were used in World War II to prevent trauma infections among soldiers. We had some antibiotics that were sulfa drugs that happened about the same time. And then we had a golden age of discovery where we had a lot of natural products, and we'll talk about antibiotics and their source from the soil, from soil microbes, that came in many waves, which we started with penicillin, then we had streptomycins, erythromycins, tetracyclines, and then iterations of those that have been modified by medicinal chemists to improve their properties and to, in some cases, get around resistance. Because one of the things that we observed immediately with the new antibiotics is you get resistance always within a period of time, sometimes 5 years. You start to see some cases of resistance arising, and we'll talk about resistance mechanisms and how we get around those with the use, for example, of cocktails of drugs. So we can talk a little bit about the resistant microbes that are of greatest concern globally. Um, uh, TB or tuberculosis is a classic that still kills millions of people, though nowadays it's more limited to developing countries or poor countries, because in the West here we have better sanitation, we have the use of antibiotics, but even with TB now we have widespread multi-drug resistance. So in some situations we have strains of the Mycobacterium, which tuberculosis, which causes TB, um, arising as resistant strains in prisons. In I think the first cases of almost untreatable TB arose in Russia, and those are they're still a threat. But fortunately, with monitoring, the TB can still be treatable, although quite challenging to treat because of the level of resistance. You often need multiple antibiotics over a long period of time
Right.
to treat. So next to tuberculosis, I think Staph aureus is one of the worst. That is much more prevalent as a problem in Western countries. So for those of you that may have
Staph
cut,
is what's
yeah,
on our skin, right?
right. So we can talk a little bit about that. So Staph aureus is colonized in about 30% of us. That means it is present in our bodies, on our skins primarily, but also the reservoir is also in our noses. And the problem arises when the Staph aureus can enter the bloodstream through a cut, gardening injury, a splinter, or whatever. And if not treated, you can get an abscess, which is the first sign. But if that isn't dealt with, you can go on to sepsis. And of course, Jackie, sepsis survivors that have been on vacation and have cut themselves, and
Mhm.
then over the weekend they find themselves in the ICU because things have gone quickly awry with sepsis. You have not just the underlying infection, but you have dysregulation of your immune system, and it can lead to organ failure and death. That's kind of the worst case scenario. So what we've been seeing over the years is an increase in resistance. And many of you will have heard of MRSA or MRSA, Staph
Yeah.
aureus,
That
because
one gets a lot of press.
yeah, it
You
is
know,
quite,
yeah, MRSA gets a lot of, uh, even in People magazine and such.
Right. And, from a personal anecdote, my mother, when she was in her 70s, had a second hip replacement surgery that didn't go well. And the implant had to be removed because of an infection. And she had multiple surgeries and ended up with an MRSA infection deep in the bone. And they never were able to get rid of it. She had oral antibiotics for the rest of her life. And of course, uh, if you have broad spectrum oral antibiotics given to you, it's going to affect your gut. It's going to affect the good bacteria in your gut that are needed help digest food, and the microbiome, so called, also actually regulates your nervous system and your well being in other ways. So to cut a long story short, she suffered from it, and once Staph aureus gets into the bone, there's really not a lot you can do. You can take the implant out, but it's a persistent infection that's problematic. So we worry about Staph aureus for that reason. In many countries, it is actually the highest killer of bacterial infections along with a few others. So there are some other bacteria that are very hard to treat, to the point where there are no other available antibiotics. They're rare, and I can list two that I'm familiar with. Klebsiella is common as a urinary tract infection, not as common as E. coli, which is one of the main ones, which also has in which resistance has arisen. But there are some strains of Klebsiella now that are essentially untreatable. They're resistant to the last line antibiotics called carbapenem, which are IV antibiotics, and colistin, which is fairly harsh antibiotic, which is neurotoxic, but is sometimes used in desperate cases. And resistance to all of those,
Mhm.
um, uh, in some cases, is is fairly common in certain contexts, and
Yeah.
mostly in poor countries. And we'll get into that, why the poor parts of the world suffer disproportionately with regard to infections and AMR in general. Mm.
Yes. And I mean, essentially, bacteria, the prime reason that we're concerned about all of these things, infections and antimicrobial resistance, bacteria have been around, I mean, forever, right? It's just that's why we have bacteria in our bodies, in our gut. We have bacteria on our skin, right? And then we have, but that's like the good stuff. And then we have the bad bacteria that can infect us and overtake things. Do I kind of have that sorted
Yes.
out in my head, right?
So let's just take another specific example. So E. coli, right? E. coli, there are benign strains of E. coli that are harmless and that are in your gut and are part of your normal microflora or microbiome. But E. coli is very diverse within the species. There are strains of E. coli that cause UTIs, and they're the major cause of urinary tract infections or UTIs. There are strains of E. coli that cause diarrheal disease as a result of food poisoning, and some of those bacteria that cause food poisoning, they express a toxin that gives you those diarrheal symptoms, which are quite severe. So you can go from healthy to a scary pathogen that has different symptoms depending on what the bacteria are expressing on their surface. They have so-called virulence factors that modulate the interaction with the immune system. And in one context, they express a certain set of proteins that allow them to attach to the bladder wall. They then invade the bladder and cause invasive infections up into the kidney in the worst case scenario. Or in the gut, they have a different mechanism to invade because of different proteins that are on their surface. Or in the good case of commensal E. coli or harmless E. coli, they don't have the virulence factors at all, and they reside perfectly happily in your gut along with all the other bacteria or good bacteria.
It's so incredible because it's like trying to maintain a balance or something so that it doesn't go all one way or the other. And then, so antibiotics, as you said, they were developed mostly during World War II, correct?
Yes, and so the classic story, which many if you probably go back to your high school or introductory biology in college, Alexander Fleming discovered penicillin fortuitously through cultivating bacteria on a dish, noticing that a mold that had grown fortuitously on the petri plate was killing the bacteria in a zone around it. And that led to the understanding that the mold, which came from a soil fungus, have natural products, the toxins that they secrete, that are part of a mechanism for competition to enable them to survive in a complex soil environment. So soil microbes, they're bacteria, they're fungi, and they're in a constant state of competition for nutrients. Most bacteria in the soil, they're critical for breaking down dead material and renewing the nutrients back as simple molecules that then can be taken up again by plants. And so in that environment, you have fungi and bacteria that are constantly trying to kill off their neighbors so that they can persist. And that has led to the discovery of many exciting and important natural product antibiotics. So these include the penicillins and then the other classes that I mentioned during the golden age of discovery. Various classes that work in different ways to selectively kill and poison the bad bacteria. And
Oh,
so, these natural products that act selectively to kill bacteria and not the host
yeah.
work through a number of different mechanisms. So there's a little bit of biology that I think we want to go through to explain that. So bacteria have cell walls, as do fungi, which are very different from our human cells. So that makes for a natural target for natural product compounds, small molecule compounds, to specifically block the growth that's needed, the growth of the cell wall to protect the bacteria and fungal microbes. So selectively killing through that mechanism is one of three main mechanisms by which antibiotics work. The other mechanism is the translational machinery. I don't want to get too complicated into this, but the ribosome is a factory within the bacterium that's made of proteins and RNA. And what it does is it translates RNA made from DNA into proteins that do everything that the cell does. We have our own ribosomes, but the bacterial ribosomes are different enough that some of these natural product antibiotics target the bacterial ribosome to make cellular proteins, but not the host cell protein. So there's a window of of potency that makes them kill the bacteria and not the host. And then finally, we have also certain antibiotics that target DNA synthesis and RNA synthesis. Again, the machinery in the bacterium is a little bit different from the machinery in the host, and that is a way that these natural product compounds have arisen as selective drugs to kill the bad bacteria.
Okay, and I definitely want to talk a little bit more about that natural product. We hear the word natural, and it creates a vision for people that it's coming from the environment. It does come from the environment, right?
Yes, from the soil.
Yes. Yes. And you're definitely you're testing me here, Robert, because I'm going all the way back to my high school biology classes when you started talking about the ribosomes and the RNA, and we had to do the matrices and all of that, or helixes. I think there was a helix involved in all that. And really fascinating stuff. And when you talk about the bacteria also, because there's different types of bacteria, which is why we have the need for different kinds of antibiotics. And the different kinds of bacteria, was that what you were referring to? Just, it sounds like they're very much in competition, sort of like a survival of the fittest amongst bacteria themselves.
Exactly. So yeah, and bacteria are incredibly diverse, and we just talk about those bacteria that cause human disease. They're two really rough classes, so-called gram negatives, which are E. coli and some of the others, but also gram positives, which have a distinctive feature, which is a very, very thick cell wall. So they stain differently. One is called a gram negative, and the other is called a gram positive because of the way that they can absorb dyes and stains. And that crude classification is important because there are, as you suggest, there are antibiotics that selectively target gram positives because they have a thick cell wall, and and the antibiotic works differently, and the gram positives are susceptible, whereas the gram negatives may not be, and vice versa. There are antibiotics that are more selective towards gram negatives. So that brings the concept of broad spectrum antibiotics, antibiotics that can kill all bacteria that are pathogenic. And those are actually quite rare now. So if you end up in the God forbid, uh, and you have an infection, they will before they diagnose the specific bacterium, they will give you a cocktail. So we'll talk about vancomycin is used
Oh, yeah.
selectively for the gram positives. For
Yeah.
gram negatives, you have the later generation penicillins called carbapenems. You put those two together. The downside is that wipes out your gut flora for a period of time, which
Oh,
will
yeah.
recover. It will recover, but then the problem is that can lead to other vulnerabilities. So you mentioned early in the beginning Clostridium difficile, which is a bacterium that in elderly people in particular, who are most vulnerable, they get C. difficile infections as a result of taking broad spectrum antibiotics. And so um, they then get diarrhea and colon perforation that can lead to
Mhm.
removal of the colon or even death. Fortunately, there've been many advances on the vaccine side. There are vaccines in development. On the drug side, there are new antibiotics that are actually selective for C. difficile and not all of the other bacteria, so that you don't get recurrence of the infection anymore. It can selectively take that out. So that this is a new approach, which I think is we can get to talking about potential solutions. The concept of more selective antibiotics, which are only possible if you have good diagnostics. And that's another
Yeah.
important topic that we can go on to eventually. So antibiotics and the bacteria that we use that are susceptible to those antibiotics, understanding what's susceptible and what's resistant is really critical to rapid treatment and successful recovery of patients.
Yeah, it's like we say with sepsis, early diagnosis and a fast diagnosis of sepsis is very important, so the patient will survive. But there's also that accuracy part in trying to determine if the doctors aren't sure. Like mine was screaming E. coli infection and everything because I had all that spillage into the gut with the colon perforation. But some infections aren't as obvious, and it's hard to determine what the bacteria is that's behind it, or the the fungus or parasite or whatnot. So they have to do like the blood culture testing and try to tease it out and figure out what kind of microbe is responsible. Because isn't it like with antibiotics, they're the key, and the infection being the doorway or the lock. You have to have the right key to unlock the right door with the infection, the key and the antibiotic and the door being the infection. You can't just jam anything into there other than starting with broad spectrum antibiotics to try to help the patient, not get sicker. But ultimately, right, you have to know what is bothering the patient, what's behind it to use the right antibiotic.
Right, and the therapies are often iterative. So you know if you have a fever and are suspected of having a potential sepsis condition, um, that there are triggers. So as you know, being on in the Sepsis Alliance organization, there are in the clinic trigger points that will trigger the use of broad spectrum antibiotics. Once maybe you have low blood pressure, you have fever, you have certain biomarkers from a serum test that suggests some kind of infection. which can sometimes differentiate just from the, the biomarkers, whether it's a bacterial or viral infection. If it's viral, you won't need the antibiotics. But when you go to the point of the antibiotics, you get the broad spectrum. And then over the next few days, it takes several days to take the blood culture, put it in a bottle to allow them to grow up to the point where you can identify and isolate the pathogen that's causing the bacteremia or sepsis. And then you test the bacteria in it with a drug panel to see what antibiotics will kill it, kill the strain. But that takes days. It takes sometimes 3 days to
Yeah.
get a blood culture positive. So at that point, once you've identified the pathogen, you can go in a more targeted way to to focus on that specific bacterium, knowing in advance what it's going to be susceptible to.
Yeah. Yes, exactly. Using like the best killer antibiotic that will kill that microbe or, take care of it, diminish it. And it's a big problem. Like AMR is a huge problem. I wanted to mention this before we kind of get into the topic of, where it impacts people the most. But how many deaths is it? Is it 1.3 million globally from AMR?
1.3 globally now. These are data from the WHO, who gets data from surveillance all over the globe from academic researchers and others. The projection is why this is such a concern is that by 1950, by 50 years from now, by 2050, sorry, 2000, by 2050, the incidence rate will go up to 10 million deaths per year. As you mentioned, now it's 1.3. It's almost a tenfold increase in global deaths from drug-resistant bacteria. But we have to put those numbers a little bit in context because um, globally, 9 million of those deaths are projected to be in Africa and Southeast Asia. And the reasons for that have to do with poor resources, and lack of access to healthcare, poor sanitation, all of those things. It's still a problem here. We're talking about the numbers, looking at my notes here from WHO uh, report, is that about 300,000 deaths in the U.S. By 2050.
Almost as many as sepsis deaths.
Yeah, yeah. So it is a concern,
Yeah.
even here in the West, where we have highly developed medical systems with relatively good diagnostics. We need better diagnostics. We need to develop new antibiotics where we have situations of untreatable infections. As I mentioned again, those rare bacteria, Klebsiella, I think Acinetobacter is another very resistant bacterium. We don't see a lot of that here, but soldiers coming back from the Middle East with war injuries,
Hmm.
Ukraine battlefields, Acinetobacter is and highly resistant, untreatable. Acinetobacter is more of a problem.
Is that what they talk about when people will mention superbugs?
Yes, so superbugs is a personal pet peeve around the use of the word bug because that refers to me as a biologist. That means it's an insect, but no.
Yeah.
So yeah, those super resistant bacterial infections. Yeah, so that exactly. So Acinetobacter and Klebsiella strains that are resistant to the last line penicillin type drugs, the carbapenems, and resistant to the vancomycins and virtually all other kinds of drugs, antibiotics that have been developed, untreatable infections, are what are often referred to in the popular media as a superbug.
Yes. Yes, they love that term. But resistant to vancomycin, now that says a lot because many of us sepsis patients are given vancomycin. That's like one of the first things they reach for, I think. And it's strong. It's so strong. They had to monitor my blood to test my vancomycin levels like every day. And I guess that's to make sure that, there's not a whole lot of that building up. But the vancomycin, I would just remember it being very strong, and it's kinda really rough on the body, I think, especially the kidneys. So if there's a
Very high dosage.
bacteria out there that is vancomycin resistant, to that powerful medicine, that says a lot. And how we're kinda getting and working our way into that topic of, what are the antibiotics in the first place and how they work, and you have already discussed this with, like, the cell walls and how they work to fight and treat infections. But how do these bacteria get so strong in the first place? I don't know
Yeah,
if
that's
I'm jumping
actually
ahead a little
no, it's a
bit, but yeah, it's
vancomycin is a perfect segue topic to talking about the contributing factors and where resistance most commonly arises from geographically. So, vancomycin resistance was, at least for the bacterium Enterococcus, which is a pathogen as well, was attributable due to use in factory farming in Europe. And until a ban of the use of vancomycin in poultry and pigs in the EU in 1997, uh, people would, farmers would throw vancomycin into the feed, and in order to, in a factory farming context, to improve growth and for the animals to do well in under horrendous conditions. Pigs packed together in pens, poultry, tens of thousands in a warehouse. Those are conditions which are a perfect storm for resistance to arise. It's the concept of selection. If you have an antibiotic that's constantly being given, the gut bacteria in the pigs and the chickens become acclimatized and actually resistant to antibiotics that's been given to them in their feed. And so you get the resistant ones survive, the susceptible ones are wiped out, and the resistance then can get spread to humans, and it gets spread to human because the waste ends up it gets transmitted to the workers, it gets into the water systems downstream of the farms, and the resistance gene known as Van A was was traced back to this problem of use in Europe. And now there are, of course, examples of many other classes of antibiotics where exactly the same thing happened. In chickens, we had tetracyclines used, and others that there was a lot of attention to trying to reduce the use of antibiotics in animal feeds. And thankfully, due to regulations, the FDA has put incentives in place and guidelines so that the manufacturers, whether it's Purdue chickens or others, to stop the use of putting antibiotics in animal feed. But that practice has not stopped in other countries. So the problem persists. And so you get the evolution of resistant microbes occurring as a result of factory farming to a great extent because those resistance genes get transferred. And this is another thing that we can talk a little bit about is bacteria can swap genes very effectively. So they can
Hmm,
transmit genes between
yeah,
each other in a
yeah.
process called conjugation, basically bacterial sex. They swap the DNA, but they also divide vertically, meaning they bud asexually as well. The key point here is that bacteria have a very short generation time. We humans, 20, 30, 40 years, maybe. Bacteria
Yes,
can be 20
yes.
minutes. And
Wow.
because a small drop, liquid drop of bacteria can have as many as 10 million present, you can imagine that in a single chicken, you've got trillions of bacteria, and with the presence of an antibiotic, you can select for a rare bacterium that might have acquired a resistance gene that suddenly takes over, and so it gets spread in the fecal matter and end up all over the place. And that's how often you get cases where you get food poisoning from due to E. coli or salmonella as a result of manure on farms getting into your lettuce or your vegetables. So it is a problem that continues to exist and does require continued monitoring and to ensure good practices in farming, but also in the food chain as the food makes it to our supermarkets. Mhm.
That is, and that's, I think it's just been a problematic thing ever since it began, factory farming and those practices. And so I think I really get it now. So bacteria reproduce very quickly, a lot faster than humans. And they're all of that, those genes get transferred just like they do with humans. We give genes to our offspring, only bacteria sounds like they have millions of offspring.
It's an exponential growth. You
Yeah,
go from 1 to 2 to 4 to 8 to 16, and within a few hours you have a bacterial culture that has as many as 10 million per ml of bacteria.
incredible. That's why we get sick so fast.
Yeah, because
And
the
and
bacteria can flourish rapidly. You have a wound infection, let's say, gut perforation. A bacteria can bloom and proliferate, and then, of course, when they start secreting toxins, that can lead to shock. And
is it like, I mean, because our audience might understand, any of you bakers out there, is it like with when you're working with yeast?
yeah, it's the same principle of exponential growth under the right conditions. Very rapid growth, where the yeast, when of course with baker's yeast, they do good things.
Yeah,
Uh, yeah, they
and
they
makes
ferment
good
make
food.
products. But even, that's funny you mentioned that in terms of food. I mean, I mentioned the bacterium Enterococcus. I used to work on Enterococcus in a vaccine program, and they're good and bad Enterococcus. Enterococcus is also used in the food industry for sausage making in Europe, but there are strains of Enterococcus that are now almost untreatable that cause human disease, particularly in the very frail elderly. So it's just another example of good and bad coexisting.
Yes. And I think the first time I learned like that there's actually bacteria all around us was making sourdough and making the starter, which doesn't require you to use yeast because
Comes
yeah,
the
the
air.
bacteria from the air. I mean, it's so fascinating. And it really is easy to understand. That one of the things I'm doing, the reasons I do some of the things I do here on the podcast and talk to scientists like yourself is science seems unapproachable for a lot of us. It feels like it's something that a lot of us can't even begin to understand. But when, as we're discussing here, it's easier to understand things when we can relate them to something we do every day, like maybe make bread or, culture yeast or make sourdough and realize there's bacteria all around us. So thank you, Robert, because that does help make it a lot more approachable for me to understand how to apply it. Okay, so we have the problems, that sort of they come from the factory farming. And are there other ways that resistance is encouraged? Like I've been told by my doctors when I get an antibiotic, and the bottle says it too, take until gone. Don't stop when you feel better. Why
That's
is
exactly
that?
right. Yeah, so inappropriate use of antibiotics, meaning that if you don't finish your course of antibiotics and take only maybe a day's worth instead of a week, what that's doing, it's giving the bacteria that you're trying to treat an opportunity to survive and the resistant ones to grow. If you don't kill the bad bacteria altogether by following through and giving the full course. You encourage the few that might be resistant to survive and grow again or recur and come back. So, yes, that is correct. So you want to make sure that you do take what's recommended, and that calculation is based on the properties of the drug. Some drugs, yeah, a single dose will be enough, and many others will require many days of dosing to achieve the desired effect of controlling that infection.
Okay, that makes me think about, for example, when I've had a dental implant procedure and I need to be pre-medicated because of my risk for infection is higher. So I am given, usually it's just a little packet. I think they call it a Z pack or some sort of a pack that you'll take 2 antibiotics the first day, and then you'll take one every day for 4 days after. But if I have something like pneumonia, I'm given a totally different antibiotic and I have to take it for up to 14 days. And they're usually huge pills too.
Yeah. No, with dental procedures, the challenge there is that your dental plaque is bacterial in nature, and some of those bacteria, they can make their way to particularly the cardiac tissue and cause endocarditis infections. So that's prophylactically you take often antibiotics through dental procedures. But if you have other risk factors and you develop another kind of infection that may be indirectly related to the dental treatment, that may require, because it will be a different pathogen, it may require a different antibiotic.
Right. Right.
Mm.
So, yeah, different antibiotics for different types of microbes and infections. And I think we touched on it very briefly. I wanted to just mention this before we go into like who exactly is at risk, especially for the resistant infections. But the antibiotics themselves, you call them natural. And penicillin came from a mold, right?
Hmm.
Like literally mold. And that's why people would joke, I think, sometimes when in my house, my husband would pull out a loaf of bread if there was mold on it. He'd say, here's some penicillin. But it's not quite that easy. But that's how I remember penicillin comes from mold. And what are the other antibiotics? Like, how did they develop all of these? How did they even, I can't even begin to imagine how they figured it out.
Yeah, it's a very interesting process. My career at Merck was really cool because we had Merck at the time had a massive screening program where they sourced soil from all over the planet, and looked for microbes that it would culture and then extract from those cultures the small molecules and then screen to see which extracts killed which pathogen. And that way look for new antibacterial, antifungal, even using the screening to identify flea and tick medications, for example, or ivermectin came out of a soil microbe, a streptomycete from a golf course from a researcher who would collect soil while he was golfing all over the place, in this case to Japan. So that was a real breakthrough for the company. Ivermectin is a very successful drug to treat heartworm in our pets, but also worm diseases in Africa, river blindness. It was used and rolled out in a widespread way, mass distribution to prevent blindness in children and adults in underserved countries. Other examples, I mentioned flea and tick medications.
I'm
These
familiar
these
with
often
those.
yeah, these often
It's our
start as
dogs.
natural products, but the chemists often then tinker with the compounds that are made on a large scale, modify them to improve the properties so that they work better in your body, they're safer, but also in some cases with the antibacterial antibiotics to get around resistance. So we have penicillin, we have second, third generation penicillins, which successively overcome resistance of the previous class. So finally, we're now at carbapenems that like the third generation, fourth generation penicillins, and we got resistance to those now. So the chemists have gone again and tried to develop another generation of antibiotics to kill those bacteria that have figured out how to avoid being killed by the antibiotic.
Wow. So the bacteria sound awfully smart, like they're always trying to figure out how to avoid their own demise by these antibiotics.
Well, they're not actually smart. It's just that they evolve rapidly, right? So
Yes.
whenever you apply selective pressure, the survivors will make it through, and because of the rapid growth and evolution, the fact that genes are swapped between different bacteria in different ways, we can get into bacteriophage, but bacteriophage, which co-evolve with bacteria, also transfer genes between bacteria as they do in humans. We have in our DNA genomes, we have relics of viruses that are still there in our genome and have been
Crazy.
important for our gene our genome evolution in general. So yeah.
Yeah. So our DNA, I mean, DNA, I think, didn't we used to think of it as something that stayed the same and was just passed down among generations. But now with epidemiology, the study of where and how these things are affecting people, it's actually that our DNA can change, right?
Hmm.
Our
Which
DNA
make
can change, and bacteria DNA obviously also change and adapt.
Yeah, at very different rates.
It's just all, yeah, and it's just all like a lot of things I did learn in science in high school and early college years. It's making sense now because that whole thing, adaptation, something when you go in the Smithsonian Science Museum and you go through the different sections and there's the adaptations. So we're constantly adapting, bacteria are constantly adapting, and it's just like a race to the finish to see who's left standing, I guess. But it's really, really cool. And it's very cool that antibiotics got their start literally from dirt. Would that be?
Many, right. So, that's the majority of antibiotics. I will say, though, that there are some fully synthetic antibiotics that are in use. I mentioned the sulfa drugs came out of German organic chemists, and there's some newer drugs like Zyvox, which is a synthetic to treat Staph aureus that came out of drug screening, and so that's probably more the way of the future. And we can get into the solutions topic and what
Yes,
needs to be done to address antibacterial resistance and newer technologies, for example, that I think will make a difference in
definitely.
addressing the problem.
Yes, and definitely. We do want to get into that solutions topic. And, just before we talk about that, there's people who, just like there are people who are higher at risk for sepsis, there are people who are higher at risk to have resistant infections. So no antibiotic will help that infection. And typically, like I'm going to have or will have 2 episodes on my podcast where I spoke on each episode with a person who has cystic fibrosis, which is, a chronic condition. And these patients in particular take a lot of antibiotics, some very long term, some for, almost a lifetime because of the presence of, constant infections, especially in their lungs. So patients with cystic fibrosis are some of the ones who are more at risk for these type of resistant infections. And I wanted to ask you, Robert, more about that. If you could expand a little bit more on that, and just talking about that it's not just patients; that healthy people too are
Right.
also at risk.
No, yeah, absolutely. So the patient populations most at risk are probably the immunocompromised. So immunocompromised patients can be cancer patients. Typically, when you get chemotherapy, it'll wipe out the more rapidly growing cells in your body. So not only will it target the cancer cells, which are growing like crazy, but also your blood cells, and your immune cells, T cells, that because they grow fast are selectively killed along with the cancer cells. So you become susceptible to infections, much more susceptible to infections. Yeah, cystic fibrosis patients are a special case or class where the problem is not being immunocompromised, but it's a genetic problem of mutations that prevent the normal secretion of mucus to be properly regulated. So you're not unable to have proper mucus turnover as a result of mutations in so-called ion pump that allows for ion transport and proper lung function. And so they are particularly vulnerable because they don't make mucus in the way that normal people do. Infections can come in and take advantage opportunistically. The other potential patient population who are at risk are people who are in the ICU. There are hospital-acquired infections that arise within the hospital because of the need to sanitize against certain strains that, like C. diff, for example, is very
Yeah.
resistant. Once it occurs in a hospital, it spreads because it makes spores that you can't really easily kill with disinfectants. You need to go to 10% bleach to really wipe it out and start isolating patients, for example, quarantining patients. Then we have, of course, then we have the healthy people who can also get very, very sick, from uh, a gardening cut where injury is a result of trauma, like or a car accident where you have damage and breakage of tissue, so that bacteria that are normally in your gut uh, can spread through damage that from
Mhm.
result from the trauma or damage resulting from wounds and so on. And we have other situations like C-sections, joint replacements are a major source. As I mentioned at the outset with my mother getting a Staph aureus infection through a hip replacement surgery. You don't have to be immunocompromised or cystic fibrosis patient to still find yourself at risk because of the more highly resistant strains that are out there.
Yeah, one of when you mentioned the lawn and your lawn work and gardening, one of the comments that sticks with me when my article went into the Washington Post about my sepsis survival, and there was a comment a lady made about, thanking us for running the topic of sepsis because her husband had an accident in the yard. He cut himself doing yard work like Saturday, and he was gone from sepsis by Monday from a cut. She said, they were blindsided by it. They didn't know really what sepsis was and didn't know that, something like a simple cut could lead to death.
Yeah, indeed. And so, yeah, in terms of improving outcomes for sepsis patients, which you and I on the Sepsis Alliance are always hearing about, yeah, there's a lot more work to be done in the research field to improve diagnostics so that we can more rapidly diagnose infections. We need better and newer antibiotics without question. And I think the challenge there, which uh, is really a major barrier, is that pharma doesn't really have the incentives to make new antibiotics. It's a money losing proposition now, and part of that is that it's really really hard to find new classes of antibiotics through the natural product screening that, for example, that we did at Merck. We were not able to find new classes of antibiotics. We kept rediscovering the old ones all over again, and although we did find some cool new ones, they weren't progressible because they didn't have the right properties. They were too insoluble, or they had other toxicities and so on. Making new antibiotics is a high risk proposition that takes a long period of time, 10 to 15 years, costs up to $1 billion now in terms of when you factor in not the early research, but where the money is, where it's costly is the clinical trials and getting all the regulatory barriers to get licensed. The other thing about antibiotics is it's that you're talking about a low sales volume because they're only used for a short period of time, and so they're not seen as profitable when they're so hard to produce. And the good news, though, is that because of the increasing awareness, there's a lot of political impetus in different countries to fund early research and to fund late stage research. And this is through organizations like the Gates Foundation, which is really focused on improving quality of life and healthcare in poorer countries, in Africa, Southeast Asia, but also nonprofit consortia like CARB-X, for example, which they get funding from governmental agencies internationally, from the U.S. government, from Germany, from Gates, and so on, and so the competitively look for new need antibiotics and therapies, and to promote them and move them forward to find new treatments, but also not just new therapies, but newer antibiotics. I'm sorry, newer diagnostics. So
Yes.
diagnostics, as we mentioned earlier, are important because you need to develop them fast. In the case of a patient that's developing an infection, you can't wait 3-4 days because you'd be dead and of course, we talked about the problems with broad spectrum antibiotics. You want to quickly identify immediately as possible the exact source of the infection. And
Right.
some of the newer technologies are really quite amazing and transformational. Think, for example, it's now possible to take human serum and use newer DNA sequencing technologies to identify just a few pieces of DNA in the blood that come from which bacteria? Well, you have a database of sequences because bacterial genomes have been sequenced for the last 10 years or
Wow.
more. You can very quickly identify even bacteria that you cannot cultivate in the lab. So this technology, there are a number of companies working on it with governmental funding. The they've been These technologies have been used in research mode clinically, and the challenge is to bring the cost down so that they can be used and implemented in a more systematic and widespread manner. So yes,
Is AI helping with any of this? Like the
yes. So
development of I would imagine.
excellent point. So yeah, artificial intelligence and machine learning is about uh, mining databases that are there, whether it's DNA sequence or algorithms to identify patterns in patient data to identify early sepsis, which I know is an important new development. But also with drug design. So there are now AI algorithms that can predict exactly how a protein target of an antibiotic will fold without using the older fashioned methods of having to make crystals and get a structure through X-ray crystallography. Now you can um, predict how a protein will fold or an enzyme that is the target of a drug, and rationally design a molecule that will block and prevent that protein from functioning in its normal way. And these technologies have moved extraordinarily rapidly just in the last few years to the point where there is room for optimism in terms of rational drug design occurring more rapidly. You still have to do the research, that you can make the predictions, but you still need the research to evaluate which compounds are safe, then the chemists will have to come in and tinker to fine tune the design to make it an effective drug that won't have off target or effects against the host that will result in toxicities. So yes, AI will accelerate drug development. It will accelerate diagnostics. Already is in both cases. And then as I mentioned, I think with political pressure, I think we are seeing funding. I think, the U.S. government is still funding a lot of research, maybe not so much in vaccines, but certainly
Yes.
in terms of antibiotic resistance.
Yes.
Back while we
And
talk about vaccines, I mean, vaccines are also an important tool to prevent infections. But
abs
the problem
no.
there is that there are many bacteria for which vaccines don't exist.
Yeah.
We talked about Staph aureus. Many companies have tried to make successful vaccines to prevent Staph aureus infections, but have failed over and over again, including Pfizer, where we had an active program, and it's because we don't really understand the mechanisms because the bacteria are so complicated. They have very complicated ways of evading the immune system, and unless you understand exactly how they do that, it's going to be hard to develop vaccines that will be effective. So that's why, for Staph aureus, we've got nothing except older antibiotics, which are okay right now if you can diagnose quickly. But we still need better antibiotics, perhaps antibiotics that will selectively inhibit just Staph aureus and not others, so that we won't destroy our gut microflora when we take those pills.
I am totally focused on, yes, keeping my gut healthy as possible. My doctors actually advise every time they give me antibiotics, they tell me to eat yogurt. Or I also found this cottage cheese brand called Good Culture that has probiotics in it too. So I don't like to take the pills so much, like the probiotic pills. I enjoy eating yogurt, but some people don't. So the probiotics are the way to go. But that's what my doctor tells me every time I'm on antibiotics, to try to, keep that gut flora healthy and balanced and not decimated by the antibiotics.
So probiotics are indeed a useful tool to help
Yes.
restore natural gut
And
flora. Mm.
I'm really glad you mentioned too about how AI is assisting, because I think it being still so new, on like a new frontier, the assumption is AI will, you don't need scientists if you have AI, but yes, you very much do need scientists if you're using AI, because as you mentioned, it's the chemist who comes in and tinkers with the formulas. The AI are like the super brain that are doing the processing, the figuring out faster, right? Than
Yes, it will
like
guide the
we could
research.
do.
It will guide the research
Guides
to be
it.
more
Yes.
efficient,
Yeah.
get
And make it
faster
more
outcomes.
efficient. Right. Because I think, as our audience is hearing, clinical trials take a long time. Discovery takes a long time, and figuring out the mechanisms like behind something like the staph infections, and how it works and how it behaves, that you need something really like a superpower like AI to sort that out or help sort it out.
Yep, right.
Now it's a global problem. Of course, we've been talking about AMR. I'm based in the US, you're in the US, but it's not just a US problem. It's global, right? I mean, it's everywhere. We're a global society.
Yeah, I mean, it's a bigger problem in poorer countries because of lack of resources there, poor sanitation, unregulated antibiotic use, lack of healthcare access, and to address that, we have a lot of funding, for example, from the Gates Foundation, who are dedicated to that, to address all of those things, better sanitation, providing better healthcare access, monitoring of resistance, and funding that research where it's happening. Yeah, the global problem arises in part from these poor countries because of global commerce. We have tourism, we have even bird migration can spread resistant bacteria across the planet fairly rapidly.
Climate change, right, can
Climate
impact
change
AI.
can impact things too, in terms of flooding that then results in more sewage being dispersed and so on, and poor health from the other extreme, which would be drought and poor nutrition. I think one of the big problems that is not understood is the impact of poor nutrition and poverty together with poor sanitation on health. These poor countries have devastating incidents of diarrheal disease that we don't have because we have water treatment plants, we have sewage treatment plants, and so we don't get cholera here in the U.S. We don't get Shigella, which are widespread in those countries.
Yeah. Yeah, it very much, very incredible how much it affects, how much it impacts. We truly have no idea until you help us start to put those pieces together of how all of these things kind of converge and contribute to this crisis. And as you were mentioning too, with, people with poor nutrition, and that's why, everybody, a lot of people ding factory farming, but more efficient ways of feeding people have to also be thought of, and to come up with solutions there, because people need to eat, human beings need to eat, and we need to have good nutrition, but we also have to get the products out there and to the people. I mean, so what I'm wondering is anyone looking into solutions to factory farming, but also being able to feed people, and You know?
Yeah, I mean, it's primarily an economic problem, right? So you can pay $20 for a dozen eggs, or you can pay $2 for a dozen
Yeah.
eggs. The $20 eggs are, certified, um, uh,
Free range,
free range
organic.
and
Yeah.
fed with non-animal products and so on. But if you're poor, you're not going to buy it. You can have to go with a $2 uh, uh, dozen. And likewise with agriculture, with soybeans and whatever other foodstuffs that are used to feed animal livestock or food for humans. To do it on a large scale to make it cheaply is a challenge, but to some extent it's unavoidable.
Yes. Yes, exactly. And I do have a chicken connection through my daughter-in-law's cousin who raises his own chickens. And the chickens are apparently very, very spoiled too. And so I get some of my eggs from them. So it all depends on, where the food is located, how we can access it, and, sometimes who you know. But as you were talking about though, there's a lot of problems. That's the state of human existence. There are always problems to solve. AMR is a big problem and we have to pay attention to it. And I like to end a little on also reinforcing what you mentioned. There is hope, right? There's hope out there.
Yes, there is. There really are some breakthroughs in terms of new antibiotics. An example is that the first antibiotic developed for a urinary tract infection in the last 30 years was recently licensed by the FDA from GlaxoSmithKline, an antibiotic called gepotidacin. Doesn't matter what it's called, but it's brand new class. And then not only that one, a second one which has been licensed in Japan from GSK, which is a late generation carbapenem. We talked about the carbapenem as the last
Yes.
line penicillin. There's a new version now which is will kill resistant carbapenem resistant strains, which will likely get licensed here too. And these drugs have been in the pipeline for a long time and have been tinkered with and have ultimately been successful. There are other drugs that are in development that are along the strategy that I mentioned early, which is selective for certain bacteria, like, for example, drug-resistant gonorrhea. There are some new drugs that are in very late stages that will selectively target gonorrhea-resistant strains in, for example, sex workers or men who have sex with men who are particularly vulnerable to sexually transmitted diseases like that. So I think we are still in a golden age of medicine, despite the concerns about AMR.
Yes.
AI is, as we mentioned, improving the efficiency of the drug discovery process. And we also touched on genomic technologies to improve the diagnostics, to more quickly identify pathogens that cause human disease. And another example, which is a bit aside from AMR, is success with personalized cancer treatments. I mean, in a subset of patients for
Oh, yeah.
sure, but
Mhm.
using genomic technology to identify from a tumor sample the mutations that cause that cancer, and then to appropriately target the cancer, which will be different from every patient, in
Uh-huh.
a way to improve outcomes for those
Right.
patients.
And yeah, I mean, and if you look to cancer in and of itself, look at how long the developments that they have made with these personalized cancer treatments, and they're mind-blowing what the doctors can do now. Because now you have lung cancer patients that are living longer than 2 or 3 years. You have patients with other kinds of tumors, breast cancer patients. I mean, there are now older women, some in my aqua classes, who had breast cancer 30, 40 years ago, and they're still here. These women are still here. I mean, because even if they get a recurrence, the current technology helps them to survive. So, I mean, there is all of you scientists, like I said, you blow me away because of the things that you're able to do, and there's always seeming like innovation new breakthroughs, new research. So we absolutely cannot lose hope here. There's what we're trying to do here is make everybody aware of this problem and, like what's being done about it. And even like in terms of our audience, knowing like what can they do as far as AMR is concerned? What can regular people like us do to help this crisis?
Well, as you yourself have done in making awareness of sepsis through the Sepsis Alliance so well, we can vote. So we can also make sure that we vote for politicians that will prioritize research and not defund it. Fortunately, with the under current situations, the Congress have upheld most of the funding for cancer and for basic research, despite the administration. So I think that
Yes.
there's reason for optimism there is that we do need to make sure that politically we vote for people that will keep the U.S. In the front line of research through the NIH and through the National Science Foundation, and to make sure that we still are leaders in globally in the field of basic research.
Exactly. And I do work with a company where, I'm on the Hill quite often. We talk about blood culture accuracy in diagnostics. And there are many of our representatives and senators who, in their little bios, they have AMR in there. That is one of their concerns. So exactly what you said, that candidates usually aren't shy about revealing their, preference and their support for AMR and, new research and new diagnostics and new antibiotics. And they're well aware that it is a problem. So it's kind of easy to figure out, who our lawmakers are who have that on at the top of their agenda. And there are quite a few. And that always makes me hopeful. So anyway, thank
Thank
you,
you.
Robert.
Thank you
I
for
get I
having
get
me.
on I get on my little tangents, especially about the Hill sometimes. Robert, thank you so much for being with me today, coming back on the podcast. I hope to have you back on again one day
My
because
pleasure.
like you're one of my
Absolutely.
favorite scientists. And science is cool and it's very important and it's not so hard to understand, when you have a scientist here like Robert who can explain it so well. So thank you again, Robert. Thank you. And it's a pleasure to have you here today. I want to also thank my audience for being here with us and being here for another episode of Laughter Through Tears and the 120 podcast. And I just want to make one last note. Don't take this as medical advice, right? It's an informational session. It is, if you have a problem, AMR, sepsis, if you're feeling ill, the best place for you to go and contact is 911, the emergency room, or your own physician. So just don't take the podcast as medical advice. Thank you,
Thank you.
everybody. Thank you for being here. Thank you, Robert. Thank you, all of my audience, for tuning in again.
Bye bye.